So good morning to you all. Good morning to one and all, both online and offline participants who have been present online as well as offline. I take this opportunity to invite our respected professor and head to share his opening remarks about the workshop.
The most respected Dean, Faculty of Agriculture, national and internationally recognized lead scientists from various industries, the advisors of this conference, Dr. V. Selvanarayanan and Dr. S. Manikavasagam, professors of entomology, director of this workshop, Dr. Selvamuthukkumaran, Associate Professor and Deputy Director IQSC, Dr. Captain R. Kanagarajan, Associate Professor in Entomology, Convener Dr. C. Kadirved, Associate Professor in Entomology, Organizing Secretaries Dr. B. Ananda Ganesha Raja, Dr. M. Ramanan, Dr. N. Muthukumaran, Dr. T. Nalini, Assistant Professors in Entomology, Dr. Prakash, Professor in Genetics and Plant Breeding, Dr. Sunil Kumar, Dr. Sathapan from Horticulture, Dr. Dhanasekaran and Dr. Jayaprakash from Education, Department of Education, all other faculty members, PhD scholars, students, and all other participants who are all watching this international conference through online. It gives me immense pleasure to welcome the chair and all other lead speakers and the participants to this international conference. Leadership is actually action, not the position. Our dean is actually proving this in all his endeavor. He is the motivational force begin this activity. So wholeheartedly, on the behalf of the staff and students of entomology, I welcome our beloved dean for this conference. At the outset, I welcome and I thank the lead speakers. They're all national and internationally recognized lead speakers from various industries. They actually accepted our invitation in spite of their busy schedule. So they are accepted to share their experiences and knowledges to the students of our Annamalai University. So I wholeheartedly welcome the lead speakers of today and tomorrow's conference proceedings. Then I appreciate the directors, Dr. Selvamuthukkumaran, Dr. Kanagarajan, and other organizing secretaries and conveners for organizing this workshop for the benefit of students. As the director of IQSC, this IQAT 21 is a very meaningful concept. So there are 4 I's in this: institute, industry, interaction, and interface. So the third I, interaction, that is most important than the other 3 I's. So the other 3 I's may be meaningful if for the third I, interaction, it is going well. So I request all the students, scholars, you kindly interact with all the lead speakers from various industries. Definitely it will give the good insight to the research or PhD research or PG research. With this remarks, I request our beloved dean to inaugurate this conference, 2 days international workshop on industry, institute, interaction, interface, IQAT 21, changing contours in pest management, which is jointly organized by the Department of Entomology and IQSC. Thank you very much.
Good morning to everyone. The Professor and Head, Department of Entomology, Dr. Aruday Nambi, the Director of this 2 days international workshop, the other organizing committee members of this workshop, senior professors from the Faculty of Agriculture, Dr. Jay Prakash, my dear student friends. It gives me immense pleasure to inaugurate this 2 days international workshop. Normally it's said that teaching without research is useless. Research without teaching is fruitless. The other one I would like to add upon is teaching and research with a vision to interact is with a good sign. That is what I would like to add upon. Because with this changing climatic scenario, the topic that they have chosen for this 2 days workshop is extremely very interesting, changing contours in pest management. Because with the change in climate, we have a lot of problems that the farmers face. One interesting thing that I had personally observed is, I'm by profession, my family by profession is from a farming family, and we've been cultivating cassava in about 2 to 3 acres. I've never seen mealybug in my life. This is the first time I'm seeing mealybug in cassava. So that's something very interesting. So I hope this 2-day workshop would really have good deliberations. It will enlighten both the institution, the industry, as well as the students would be benefit much out of it. So thank you very much for giving me this opportunity to inaugurate this 2 days international workshop on changing contours in pest management. Thank you very much.
Thank you, sir. I thank our beloved Dean for inaugurating this workshop. And it's my time to brief the audience about the aim of the workshop. So, our most respected Dean, Professor Head, my Professor, Dr. S. Manikavasagam, Professor Prakash sir, and my colleagues from the department and from other departments, students, online and both offline. Good morning to you all. I take immense pleasure to brief the audience about this workshop. The event was conceived with the idea of conveying latest industrial trends to students and academia to equip themselves. That is the main idea behind organizing this workshop actually. With this aim, we have invited many national and international lead speakers from various industry majors with the aim of sharing their knowledge of ocean with us. So with the readiness, they all accepted my invitation, and almost around we have 10 speakers with us in the 2 days, and it is jam-packed session. We have 4 technical sessions, forenoon and afternoon, each for 2 days. So having 2 speakers minimum in each session. So tomorrow forenoon session has 3 speakers, and it is supposed to be a brainstorming session for the 2 days in continuance. So I take this opportunity to request the participants to immerse deep in this ocean of knowledge for these 2 days and get themselves enlightened with the latest trends that is happening in the industry and the future thrust that is going to decide the next decade. So I thank one and all for participating, and I request a sincere participation from the participants, because it is very hard to get all the speakers, because they what told me once I am calling them and asking them for a date, what they told me is that they readily accepted, but the only hiccup is that whether they are traveling by flight or not by that time. That is the only hiccup that they had. And once that is over, they are ready to share their knowledge immediately. It is a hard thing to happen, And I request all the students, scholars, as well as academia to use this opportunity to interact with the industry lead speakers and get themselves enlightened. Thank you. So with this, we will be directly going in for the technical session. And in the morning, we have two speakers. The first speaker of the day is Dr. Bhairappa, lead entomologist from Geogen Technologies Private Limited, formerly Pest Control India Limited. He will be presenting a talk on decoding insect communication. Already he is online, and I request the participants a few minutes to make arrangements for the presentation. So I request Dr. Bhairappa to share his to be online. So we have been making you co-host. Yeah, thank you, Bhairappa. Once again, please bear with us. We will be making the screen ready so that you can start your presentation. I request Dr. Ananda Ganesaraja, the organizing secretary, IQAT, to introduce the lead speaker, Dr. Bhairappa.
Welcome to the technical session. In this sessions, we have two lectures. One on insect communication and another on residue management. Dr. Bhairappa is a lead scientist, entomologist in Geogen Technologies Private Limited, Pest Control India Private Limited. Third, agriculture and MSc entomology in the University of Agricultural Sciences, Bangalore. Got his PhD from University of Levita. It postdoc. Is lead scientist entomology in Geogen Technologies Private Limited, formerly Pest Control India Private Limited. It is BSc Agriculture and MSc Entomology in the University of Agricultural Sciences, Bangalore. Got his PhD from University of Lelita. It is postdoctorate University of Cincinnati and Zeg University of Life Sciences. Published full-length research paper in peer-reviewed journals of high repute like Science Advances, Journal of Insect Physiology, Journal of Chemical Ecology. Supervised and mentored many student research. We welcome you, sir, to deliver a lecture on insect communications. Welcome to all.
Am I audible?
Yes, sir. You can start the presentation. We have been projecting your screen on the TV.
Okay, sure. Let me know once.
Presentation. Yes, that will be present.
Okay, yeah, thank you. Well, I guess for the better bandwidth, I'm going to turn off my video for now. Okay, so morning, dear colleagues, research scholars and students, and thank you, Dr. Selva and team, for organizing this 2 days online workshop on changing contours in pest management. In spite of these pandemic times, and it's kind of like difficult to kind of like pull this diverse set of plenary speakers to continue our learning in the field of entomology. Again, thank you for the invitation to share my views on how the Chemical Ecology Foundation was laid and the current progress that we have been making in insect communication using semiochemicals with special emphasis to pheromone. That's what I will be talking. And firstly, it is an honor and pleasure to kind of like reconnect with the alma mater. I'm also part of the Annamala University. I do kind of like recall some of the names by the times I was there in 2010. So the idea of using species-specific behavior modifying chemicals for the management of like the insects in agriculture, horticulture, forestry, stored insect pest, and the insect vectors of diseases has been a driving ambition through last 6 decades of pheromone research. So in the last six decades, hundreds of pheromones and other semiochemicals have been discovered that are used to monitor the presence and abundance of insects so that we could protect plants and animals against the insect pests. Give me a second. Yeah. So if you look at the progress that has been made from the identification of first 6 pheromone, which is Bombicol in silk moth by Bitternet and co-workers in 1959, to the identification of olfactory receptors in Drosophila by Leslie Wolschel lab, and also further the identification of olfactory receptors in the silk moth by Krieger and co-workers in 2005 is very spectacular. So pheromone communication, including the generation of behavioral responses, is now being dissected at a molecular level at different labs around the world, especially if I have to mention this one in Benton in Switzerland. So our imagination, I believe, is not sufficient to envision the knowledge in insect olfaction and chemical ecology in the next 50 years from now, provided that researchers have the mandate and the resources to carry out this work. So the idea of chemical communication was not new, even in 1959. That's when the Bombicol was identified. So there is enough literature that suggests, for example, ancient Greeks knew about the secretions of female dog in heat that attracted males. And then Charles Butler had warned the feminine monarchy, that's in the book, that if you're stung by a one-honeybee bee, so there is a possibility like you are kind of like attract where there will be other beings kind of like other bees will be following you. So that suggests there is some kind of communication even in the olden days. And also Darwin also suggested, so they use these chemical signals alongside visual and auditory ones as outcomes of sexual selection, describing the success of the smallest among breeding male crocodiles, ducks, goats, and elephants. However, I guess the most important one is like close to what we even, the one we follow currently is by Jean Henry Fabre in 1870. How male emperor moths flocked around a female moth hidden behind a wire gauze, but ignored visible females sealed under glass. Surely it's an indication that her smell was the attraction. So we know that the Carlson and Lucier were far-sighted, noting that pheromones were likely to be used by a wide range of animals, including fish, underwater crustaceans, as well as land animals and insects. So they predicted the most pheromones would act via conventional senses of smell or taste, but that some might be ingested and act indirectly on the brain or other organs, as it happens in termites whose pheromones affecting caste development are passed down by mouth through the colony. Carlson also kind of like interested, given his interested in the theme of kind of like in the sense of biology, he also catalyzed a completely new field of study in biology by asking a young biologist neighbor, that is Dietrich Snyder, if he could invent something in order to kind of like assess what the Bombical, what the Witten and co-workers identified. So that's when the Dietrich Snyder came up with the electroantenography, which we kind of like the electroantenography, we still use it today. So basically what we do in the electroantenography, we kind of like connect two wires at the both ends of the antenna and kind of like measure the electric signals when stimulated with different volatile extracts, for example. Further recordings and then advances have been made to record the activity of single antennal sensory cells, followed in later years as mass and their pheromones become a key model system in the neurobiology. So in the last 60 years of curiosity-driven pheromone research have yielded a profound understanding of sexual communication in insects. So the discovery that minute amounts of species-specific chemical stimuli encoded by discrete receptors on the antenna instantaneously elicit a conspicuous, often flight. Our orientation behavior has been a source of inspiration for fundamental research on the insect olfactory system. So currently, the research ranges from biosynthetic production of sex pheromones to peripheral perception by odor and receptor neurons, and then central processing of the olfactory input and the resulting behavior. So that's the kind of like way we stand right now. So if you look at the perception of odorant, which is very essential for an animal to identify relevant food sources and suitable sites for egg laying, and also to avoid potential threats, which is very true in the case of insects, where the precise discrimination, interpretation of what is an essentially unlimited spectrum of chemical information impacts the numerous behavioral decisions that directly contribute to their success, and in many cases, its survival. So insects in general use sensory appendages such as antennae, maxillary pulpi, and sensory hairs on their legs, mouth parts to detect thousands of secondary metabolites that are released by the plants. A closer look at the Drosophila melanogaster, we can see like several sensory appendages are equipped with a sophisticated olfactory sensory system. Here you can notice like on the antenna and even on the maxillary pulps. So I have kind of like coded them as like whatever the literature we kind of like follow it. A stands for antenna, B stands for the sensilla. So B is basiconica, T stands for trichoidea, and C is siliconica. There are other different types, like for example, Auricelica. So every group of sensilla have different roles when it comes to the functional behavior. For example, here, so the trichoidea sensilla are mainly involved in the detection of pheromone molecules, and basiconica are mainly involved in the basic food odorants. However, when it comes to siliconica sensilla, they detect acids and amines. So that's an indication they could kind of like differentiate like acceptable, like palatable food versus the unpalatable food when it comes to that. And they also have receptors to detect humidity in the nature and also the temperature changes. So we think like insects might just kind of like moving around, but once you kind of like delve into the system itself, we get to learn a lot of things that is translated into medical research as well. So to interpret these signals, insects utilize a range of chemical molecular components that center around several large families of chemosensory receptors, which are housed in a diverse array of hair-like structures that I mentioned sensilla. So these are non-randomly distributed across peripheral appendages like antenna and palps. So the number and type of sensilla present on the chemosensory appendages vary according to species as well as their developmental stages. For example, if you consider larvae and adults, there will be a lot of sensory hairs on the adults compared to larvae, which would be very limited, would be mostly limited to like very few numbers, maybe 10 or 100 maximum. And also the numbers differs with the gender as well. While it remains unclear how sensilla specific ultrastructure contributes to the functionality of each sensilla type. The high degree of diversity may reflect significant evolutionary consequences for sensitivity in odor trapping and phylogenetic and our developmental constraints evolved with physical environment. So the discovery of olfactory receptors in 1999 awarded Nobel Prize jointly to Rickard Axel and Linda Buck, Linda Buck was the postdoc in his lab. So knowledge on olfactory receptors help the researchers to identify or to understand the insects in both ecological and evolutionary perspective. And another group of receptors is inotropic receptors. So that's the ones, inotropic receptors are mainly housed in the coeloconic sensilla. So there is not much information available because these were recently, like a decade ago, these receptors were identified. But when we look at the evolutionary perspective, so IRs come in the lineage first compared to the ORs because they're mostly conserved. Whereas if you look at the evolution of insects, they become kind of like from generalist to specialist or specialist to generalist based on the host availability, where they need to kind of like change their host finding, especially with the Basiconyx and Trichoideum, just to kind of like avoid the cross contamination within their group. So you can see like a lot of pores on the sensory, on each sensilla type. So these pores will help the odorants to diffuse, for which they enter through aqueous lymph that must be traversed in order to reach the spectrum of the molecular receptors present on the dendrites of olfactory receptor neurons. So this is a schematic olfactory system in Drosophila, where you could see like a lot of sensory hairs on the antenna, like that's the periphery. And then as you kind of like go deepwards, there is a congregation or like there is a convergence happening both at the primary and the secondary centers. So the mostly odorants are discriminated by combinatorial coding as olfactory sensory neurons expressing the same protein receptor converge onto the same glomerulus within the antennal lobe. For example, all the red coding will converge into one glomerulus, and mostly the pheromones are kind of like converging to macroglomerular complex. That's the major chunk in the antennal lobe if you look into. So the activation of diverse set of chemosensory receptors by odorants provide a range of both excitatory and inhibitory inputs for the generation of olfactory receptor neuron action potentials that are further processed across several layers of downstream neural pathways. So at the same time, the timely termination and desensitization of peripheral signaling is required to promote the sensitivity and selectivity of olfactory receptor neuron-based signaling. Although the precise elucidation of the underlying mechanism responsible for this component is still undefined. So until 8 years, we believed that olfactory binding proteins play a role in transport of most of the odorant molecules to the receptor. However, a study in Drosophila showed that for one receptor, even with the absence of olfactory binding proteins, it's still going to like elicit the excitatory or inhibitory response. So that's where we stand. And that, so before I kind of like get into the advantages or like where our pheromone research has kind of like led into applied research. So currently there have been studies to look into, for example, so just to kind of like go back, when Buchanan and co-workers identified the pheromone, they used lakhs of mass just to identify, just to kind of like get 1 milligram of sample. However, we have a sophisticated techniques right now. Currently, we could use one silk moth is enough to identify what the components are and what is the role of each thing. So many who began pheromone research in the 1960s, were influenced by the concept of integrated pest management that were coined by Stern and co-workers in 1959. And in addition, Rachel Conson's plea for biorational pesticides in her novel, Silent Spring. Incidentally, the use of synthetic pheromones for environmentally safe insect control was postulated soon after the discovery of silk moth pheromone, well before the pheromones of economically important insect pests were known. So research scientists who in kind of like 1990 beautifully phrased, there are many other reasons for using pheromones. So one of them is that they're elegant. I completely agree with that, with all. And three other main elements accounts for the fascination of insect pheromones and their feasibility for insect management are, because we know that pheromones are species specific, most of the cases. And they are active in very small amounts, and the vast majority are known to be non-toxic to animals. So a group of researchers in Israel kind of like working on if there is a possibility for developing resistance to pheromones in mating disruption fields. However, we do not have like very clear view on that. I would be surprised to see if there is if they happen to prove insects do develop resistance to pheromones, which is very unlikely. Given pheromones are very species specific, since the discrimination of conspecific and heterospecific pheromone signals is a key element in the evolution of specific mate recognition system in many insects. Even synthetic and incomplete pheromone blends usually affect only the target, with the possible exception of taxonomically closely related species. Insects use extremely small amounts of pheromones for communication. For example, the calling female of codling moth, which is Cydia pomonella, release a pheromone at a rate of several nanogram per hour. If we compare the same thing to the secondary metabolites released by the plants, for example, in apple trees, so one of the main volatile component is the farnesin, racemic farnesin, which is estimated to release at the rate of like several grams per hectare per hour. So we can kind of like get an idea like why insect olfactory system or the sensory system has to be precise, while since they have to identify the odor cues at very at nanogram or even the picogram levels, given the how much dilution is happening in the real time and the environment. So if you look at the practical uses of mass trapping in brinjal, so I'm kind of like jumping into application part to see, I mean, we know that insects, pheromones have been used in monitoring, detection, and then mass trapping, and then the mating disruption. So if you take an example of mass trapping of fruit and shoot borer in brinjal, we are aware that severe losses are caused by the fruit and shoot borer. In South Asia itself, brinjal is one of the commercially produced by approximately 7,000 farmers on 570,000 hectares in India alone. Whereas in Bangladesh, 40% of all vegetables produced are eggplants, providing farmers with a regular year-round income. We know that insecticides appear to be largely ineffective for control of this pest because the protection offered by the fruit itself and the other cause is insecticide resistance. So after the development of pheromone-based mass trapping strategy and optimization of the pheromone blend and dose, trap design, and placement to field implementation, mass trapping without the use of insecticides has led to 50% and higher increase in marketable fruit, which has been attributed to the combined effect of mass trapping and enhanced impact of natural enemies. Additionally, we could also see like the secondary pests such as mites, whiteflies were reduced in the pheromone plants given the less exposure to the chemical molecules. So given estimated sales of pheromone leaves reported in India and Bangladesh, at least 15% of all the farmers have now adopted the pheromone technology, and this is increasing year by year. So here, this is the information I got it from Rizgal paper. It's kind of like beautifully phrased, like kind of like compiled all this information. And then if you compare the doses of pheromone used between mass trapping and mating disruption, lures used for detection and monitoring release typically 10 to 100 times more than the calling female. Like I mentioned in the previous slide, females typically release picogram to nanogram level per hour. Whereas when it comes to mass trapping, so we end up using like 10 to 100 times more than a calling female. And the mating disruption dispensers might release up to 10,000 times of pheromone here in the, for example, cardamom, which amounts to release rate of like 10 to 100 milligram per hectare per hour. So currently we have like different technologies for mating disruption. One would be using the hollow tubes, and the other is like, so once you kind of like set in the hollow tube, the problem is the pheromone is continuously released into the nature 24/7, whereas the current technology available is we have the time-based sensors where the aerosols are released only during the active period of the moth activity. So in this case, we could enhance the field activity of a given dose. For example, 100 milligram or 100 gram might last much, maybe like 50% or 60% higher than using a hollow tubes. So the seasonal application rate of cardamom for mating disruption of codling moth in orchards is up to 100 gram per hectare. That's the current recommended thing. And worldwide annual production of cardamom is estimated around 25,000 kg for codling moth control in approximately 21,000 hectares. So you can see like from 1990s to 2010, there is an increasing trend and the amount of pheromone we are using, at the same time limiting the use of chemical molecules. So this is another case of adoption of pheromones for the control of grapevine moth, Lobesia botrana. So there were experiment trials conducted in vineyards to area-wide campaigns by involving plant protection entomologists, growers associations, and pheromone industries. The coordination of mating disruption field campaigns is a complex task. I agree with that. So indeed, despite this complexity, approximately 1,500 growers participated in Northern Italy in 1999. So by the end of 90s, mating disruption had led to an area-wide reduction in population densities, and it became widely accepted by growers in Germany, Switzerland, and Northern Italy. A challenge in these European vineyards is now to develop novel strategies against new pests that are not affected by the mating disruption, especially leafhoppers that vector plant diseases. Meanwhile, European grapevine moth has been found in Chile and California. In Chile alone, these 40,000 hectares are now under mating disruption in an attempt to eradicate the newly established population. So given the idea of replacing hazardous insecticides with environmentally benign and species-specific odorants is still a current research challenge, but the emphasis is shifting. The motivating force for green sustainable insect control is no longer merely the health of rural workforce or the safety of agriculture and horticulture products, nor even the attempt to promote organic farming. The matter is more urgent than it was 50 years ago, and our concerns are the necessity of establishing sustainable insect control methods in times of increasing food security. So currently there are incidences by Sweden and German groups to come up with a plant that produces pheromone instead of we setting up pheromone traps and lures for mating disruption. So that's the current status of pheromone communication right now. I'm kind of like, I didn't want to kind of like overboard with the information rather than a discussion like the dean suggested. So I would be happy to kind of like discuss, open to all your questions. And these are the job alerts. Maybe some of you might be interested to apply. Keep an eye on these positions. Yeah, I would be happy to answer any questions.
Thank you, Dr. Bhairappa, and we request participants to raise their questions through chat box, and we will be in a position to forward it to Dr. Bhaira so that he can answer. Participants, if you have any queries, please raise through chat box. So Dr. Bhaira, our dean, has raised a query. Use of traditional formulations like Panchakavya, right? Does it affect the olfactory system of the insects? Pest, especially the pests.
Mostly in the mating disruption, well, most of the pheromone technology is like very species specific. Since this Panchagavya and all these kind of like plant origin, which is very abundant in the field already. Most of the compounds, if you take an example of grass, they're like acetates and alcohol very abundant. And most of these Panchagavya and all these products, the volatile profile may not be different from what we already have in the nature. So I believe it's not going to affect the pheromone efficacy in the field condition. Unless if they are using some deterrents, for example, chili-based, because these volatiles act as repellents in the field conditions. Yeah. I see there is one question on Iselvam, like what is the current success rate of pheromone technology in India? Well, to answer to this question, recently CIBRC has approved mating disruption for cotton bollworm, pink bollworm. So we are kind of like picking up, but it's going to take another 10 years to completely come into a state where the Europe and Northern America, where we're going to like use a lot of pheromones in order to kind of like mitigate the problems.
Dr. Bhaira, Dr. Sivakumar from ICR has raised two questions. Can you clearly differentiate between mass trapping and mating disruption? And he also asked about what about the pest developing resistance against pheromones?
I think I will answer the second question first. To my knowledge, and even there was in 2018 in the conference, there was a presentation where researchers were talking about, so there is an evidence insects are developing resistance to pheromones. Apparently, it's very hard to prove, but nothing is proved in the lab scale, which is kind of like easy in case if there is a resistance. And to differentiate mass trapping and mating disruption. So in mass trapping, basically, you are kind of like directing or orienting males to a point source of release, where you could kind of like have a trap and kind of like trap them into a plastic bag or whatever you want. Whereas in case of mating disruption, you are confusing the conspecifics not to find the males or females, whichever you're targeting. Can we attract parasitic Hymenoptera into field after using this technology? Yes, we could attract, but in this case, you would be using pheromones. There was a recent study where they have shown instead of attracting or instead of controlling the main pest, you would use some chemicals or the naturally originated chemicals to attract the parasitoids into the field.
Bhairappa, good morning. This is Manikavasagam, Professor of Entomology. Can you throw some light on the next generation biological control using pheromones as just raised by Dr. Krishna Chaitanya? Anything in India, we have any alamones or chiromones for parasitoids or predators?
As such, not as a large practice, we do not have any chiromones for natural enemies. But especially if you ask me, like use of pheromones in the mass trapping, since pheromones would help you to trap the females as well. So Pierre Ester is one of the success story by one group in USA where
Can you repeat it, please?
there is one group, Pierre Ester.
Okay, okay.
Pierre Ester for the control of codling moth. So when you combine, it is found to be, so that was like by accidentally they found this compound. So they had this 2000s of compounds they had sitting in the lab. So they wanted to screen all these compounds on the antenna through electroantenography. So apparently they kind of like found it. They had like no idea it's going to work. So that's the only compound so far is very effective in the field condition, although it has a plant origin. And when we look at the number of receptors dedicated to Pierre Ester in codling moth, there is like a lot of them. I can say like 20 to 30% of the Basiconyx and Auricelica sensilla are dedicated to Pierre Ester. So that defines the importance of this compound for codling moth.
Thank you so much. Thank you.
So I request the audience to post their questions so that we can, Dr. Bhairappa can answer. So meanwhile, I just request Dr. Bhairappa to introduce our students about the SSR technology. So and its role in pheromone research. A brief outline about the SSR.
Yeah.
Because one second, Dr. Bhaira, because Dr. Bhaira has been working with SSR almost for the last decade. He has been well versed in single sensism recording. And if you could throw some light to our youngsters, so that will be a motivational point for them to take up further research. Yes,
Yeah,
Dr.
sure.
Bhaira. Yeah, please.
So you might be all aware of the electroantenography. So the main difference between electroantenography and single sensitime recording. So the information, whatever you get from the electroantenography is a collective response of all the neurons that are present in the antenna. So the response could be from pheromone cells or plant cells or acid cells, or could be from humidity or the temperature. Whereas if you shift, instead of recording from the all cells, the resting potential, so you are measuring the action potential in the single sensitime recordings. The advantage is, so you get to know how sensitive or how specific this compound is. So it's not only, so you kind of like come up with a compound, you see like how many receptors are dedicated for a given compound. So based on that, let's say, instead of using pheromones, I would throw a compound that might repel all the insects from the field. Because the idea is, I will take an example of Grapholita molesta. That's my PhD model insect that I use. So a study by Tom Baker in USA, where he showed that even in the behavioral studies, if you separate the two sources or two compounds by 1 millimeter distance, so the insect is detecting, so this is not the compound or the blend I'm looking for. The reason is, if you go back to the electrophysiology, so these two compounds, for example, each sensilla houses multiple receptor neurons. And in this electrophysiology, we found out that one neuron responds to major compound and another neuron responds to minor compound. Whereas in case of Grapholita molesta, like I mentioned in the behavior, these compounds, even with the separation of 1 millimeter, they could distinguish or define this is a completely different blend. So it suggests you like how important are the minor compounds And also, once you identify these compounds, these can be translated into, once you know the significance of each compound and how sensitive it is, and how fast this compound has evolved through bioinformatics. You could do the RNA sequencing. That's the next step once you do the single cell recordings, where once you identify a receptor that is responsible for a given molecule, it is very difficult. For example, let's take an example of cockroach. If you want to, and if you take a look at the antenna, there are like thousands of sensilla on the antenna, and it's very difficult to know which receptor is responding to which compound. So once you know the receptor through genomics, you could take out that receptor and express in Drosophila basically. So basically you're taking a receptor from cockroach and express the same thing in Drosophila, which is well known. We kind of like know millimeter to millimeter how Drosophila responds. So I mean, that's the advantage of genomics tools we have. So I see a couple of questions. Do we have pheromone technical manufacturer in India? If no, why? To answer, yes, we do have technical manufacturers in India, and Gaijin Technologies is one of them. And how the ecological factor influence the pheromones in field conditions? I guess that's a good question. Let's say if you look at the environment, we know that insects, most of the volatiles are odorous compounds are in the nature. If there is low temperature, you can notice that in the winter times, there is not much insect activity. The reason is like volatiles, they're not so evaporative under low temperature conditions. I guess I will go back to one electrophysiological story in French group, where a student was conducting experiments and he didn't realize the change in temperature was happening between morning and afternoon in the air conditioning. So the recordings that he made in the morning were different from the record. The sensitivity changed. The response, olfactory receptor neurons response changed from morning to evening when he compared. Then he kind of like logged into the temperature. And that's when he found, so the change in temperature, because going from 18 degrees Celsius to 24 degrees Celsius, the volatility of any compound would change. That's like a thumb rule. So these environmental factors, yes, it doesn't matter. For example, if there is an insecticide spray taken up, and if you install a trap in first week or 2 weeks after spray, and yes, it's going to affect the pheromone efficacy in the field conditions. I hope I answered the question. So another question, if you use a variety of pheromones together in the field in a single trap, what is the effectiveness of the trap? I guess it goes back to the basic question or the basic definition of pheromones. They are very species specific. So once you combine like two compounds, what happens is like, it might work for only few insects. For example, if you combine Grapholita molesta and then the codling moth, it's very likely you might capture more codling moth and very few Grapholita molesta, but you are wasting the resources, knowing that insects are very species specific. And if they attract or not, I would say most of the time these blends would become repulsive. Let me see.
Dr. Baira, there is one question about, is there any mixed attractant or pheromones for attracting or trapping different insects? Asked by Janagar.
Just answer the question.
Answered, huh? Sorry. I just lost track of it.
No problem. Pheromone traps work will. Okay, that's good to know. Rhinoceros beetle pheromone loops are working.
Okay.
request to unmute for few clarification. What is the function of odor and binding protein in olfaction? How to bind on the receptor site? So that's basically like a lock and key, key and lock system. That's from Mr. Sarkar. So the thing is like, this is like a more of a key and lock chain system. When a odorant molecule enters through the pores of the sensory, through a sensilla. So there are so many odorant binding proteins present in the sensillar lymph. So basically they transport these odorant molecules to the receptor site. So that's the function. Is there any method related to control wax moth through pheromones? Yes, I guess. The identification of pheromones in wax moth has not been so successful. I guess there are a couple of compounds that we could still use it. How does the weather parameters influence the effect of pheromone traps? We already talked about it. Basically, when there is a higher temperature, the pheromone evaporates faster. If the temperature is low, the evaporation becomes slower. It goes the same for the humidity. Higher humidity, the distance traveled by volatile molecules is shorter compared to the dry climate. Any possibility for whitefly free field biologically? So there are a lot of difficulties to come up with pheromones, for especially for besides Coleoptera and Lepidoptera. You might have seen a paper on locust recently by a Chinese group where they identified one compound. I mean, it's large scale trials that still has to be conducted, we will see. But apparently the number of molecules available for hemipteran insects are very limited except thrips, if I recall right, and aphids. And again, they are species specific. Aphrodisiac pheromones, what its role in IPM? I guess aphrodisiac, basically they're sex pheromones. And the role is pretty clear. Like they do have a large influence in the IPM. Why are pheromone traps are not getting that much recognition as they should? I guess there is a lobby. And again, there is unawareness by the farmers from the farmers level. As a farmer, I mean, myself coming from a farming background, when you do not install the traps at the right time, and a female has already laid eggs, there is no way you could use any pheromones or any pheromones to bring down the population. More of a awareness and then patience to use the pheromones. Anti-aphrodisiac pheromone in IPM, not much research has been done, but again, there are very few molecules that might work in the field. Can pheromones completely replace conventional pesticides? Will the growing market of pheromone affect the conventional pesticide in use? Completely replace, I would say no. Given these pheromones are species specific and a crop is infested by multiple insect species, and we don't have pheromones for all the insects. Growing market. Yeah.
Dr. Dr. Suresh Kumar from Razi Seeds is waiting for you, Hussein. Dr. Suresh Kumar. So, okay. Dr. Kadirvel, Associate Professor for Entomology, is posing your question.
Dr. Bhairappa. So I was very much satisfied and I was very happy about your presentation. I'm the convener of this workshop. I would like to ask a small question. So how do these pheromones will be affected by the ecological factors in the field condition, because we are using different kinds of pheromones for the control of various insect pests. How do the ecological factors will affect the efficiency and the effectiveness of the pheromones?
Well, I guess if I understood right, using multiple pheromone blends in the field, in the same field. Am I right?
So not clear to me.
Okay.
Pardon?
Yeah, if your question was using multiple pheromone blends in the field, same field condition, that's not going to affect anyway, because given the most of the pheromones are species specific. Again, for example, let's say in Spodoptera, some species might share the same compounds, but again, if they are not in the correct ratio that are biologically relevant, it's not going to affect the other species.
Okay, thank you.
Sorry, appreciations to Dr. Bhairappa. This is Suresh Kumar from Rasi Seeds.
Yes, please.
Yeah, appreciation for impressive and interesting presentation. And a few clarifications. If you can brief on the nanotechnology based slow releasing pheromones, which is being introduced recently, one thing. And second, this mating disruption technology we are using for the past few years on the trial and error basis for pink bollworm resistant population. Already pink bollworm has developed a resistant across the country since 2017. So we are trying both mass trapping and mating disruption technology. As you rightly said, the complexity of the mating disruption technology is that the coordinated effort. Either we can go only through the community approach, a village-based approach. So that is a big bottleneck in adopting the technology. Is there any evidence that a minimum acreage, any 5 acre, 10 acre, 20 acre, we can do effectively this mating disruption technology with your experience? Only two questions. One on the slow releasing pheromone, and second on the minimum acreage we can adapt for the mating disruption technology.
Yeah, thank you. So the thing is, like for the nano-based technology, yes, there are efforts are going on. To my knowledge, I haven't seen what happens is like when you kind of like slow down the release of pheromone, then you are kind of like missing the long range insects from the long range. And trials, like small scale trials for mating disruption is possible. It should work. I wouldn't see like why it would be unsuccessful. Yeah, 5 to 10 acres, yeah, it should work. And again, you need to kind of like use the correct dosage. So where we will get the basic training on how to extract, identify the pheromones, handling of electroantennogram and JCAD. Well, Dr. Krishna, apparently there are very few labs in India. One is NBIR, IICT, IIHR, and NCBS. So these are the 4 institutes where you could approach them to learn these techniques. I'm afraid that they may not encourage, given you are going there just to learn the technique. See if there is a possibility you could collaborate on something which would be mutually beneficial. We do have these setups in our institute, since we are a private organization. Unless there is a project that comes up and the expenses that are covered, it's very unlikely the top management would encourage this one at the private sector. Yeah, these are the 4 institutes you could consider: IICT, IIHR, NBIR, and then NCBS. What are all the carrier used for lure? Basically, it's a solvent. Yeah, any solvent. For example, you could use hexane, acetone. If you want to make the release rate very slow, you could use some oils. Explain the role of pheromone in pest in stored best of stored products, why it is not practiced that commercially while comparing with field pest. The problem is like mostly these are stored in drainages and the ventilation is not that great. Even if you use the pheromone, if there is no evaporation and kind of like the spreading of the pheromone happening in the drainages or the storehouses, the efficacy is very low. However, for Lepidopteran, people still could use. It's likely that people are not aware of it. And then again, when to install, that question needs to be answered. I guess one of you, since some of you might be doing your master's or PhD work, if you have recently started, try to, I guess the problem is like the hosts availability or this test subjects availability. One thing could be like field evaluation of pheromones that has been identified for locals. So maybe give a try for that and see if that's going to work. Markedly available pheromones are originally extracted from insect or purely artificially synthesized. I guess it's chemically synthesized. Yep. Because from each insect you would get hardly 20 to 30 nanogram, even if you extract. Where we get, I guess you could approach some of the manufacturers. Apparently, most of the manufacturers, especially the private, may not sell in small quantities. Perhaps if you know the compound, approach some chemistry labs, they could help you. Please repost your question in case if I missed some of yours.
So participants, if you have any more queries, please raise it up in chat box.
What is the success rate of aphid trial and sex pheromone traps? Why it is not popular as like Lepidoptera and sex pheromone traps? So the reason is aphid pheromones are like racemic. When I say racemic, these are hard to synthesize. Sorry, isomers, not racemic. These are like isomers, hard to synthesize. But again, they are very volatile in nature. The moment you bring into the field, it's very likely, let's say if you load 1 milligram and maybe in 3 or 4 days, that might be reduced substantially. And since pheromones are aimed for like alate insects, how many of the aphids are alate in a given colony? So that something needs to be considered. Because nymphs can't fly into a trap.
So if no more questions are coming up, I request Dr. Anand Ganesh Raja to propose vote of thanks to the lead speaker, Dr. Bhairappa.
Thank you. On behalf of Department of Entomology, I would like to thank Dr. Bhairappa for his brainstorming lecture on insect communications. He emphasized the insect communication by using olfactory in Drosophila and also mass trapping strategy by using pheromones in different crops, in particular mating disruption in codling moth. Finally, he concluded that use of pheromones will pave a way towards green solution in insect pest control. Thank you, sir.
Thank you so much. And I wish you good luck with the rest of the talks. And I hope you make it and get as much information you could. Thank you, Dr. Selva, again. Thank you so much.
So thank you, Dr. Bhairappa, and
Yeah, if there are any questions, like
So
any
the
specific
next-
questions related to academia, you could share my email ID to them. Happy to answer. And in terms of like how to approach a abroad lab and what factors that needs to be considered.
Yeah.
I would be happy to come down once this post-COVID times and maybe give a lecture on abroad opportunities in person.
Thank
Yeah.
you. Thank you. It's great gesture, Dr. Bhairappa, and we will utilize your expertise soon. Once the pandemic is over, we will be asking you to be physically present here and guide our students for abroad opportunities, because openly being ready to share those things, it's a great opportunity for these people who have been from a rural background, right? Thank you. Thank you for your gesture, Dr. Bhairappa, and I will certainly convey to this to our President and the Dean so that it will be used by departments. Thank you. Thank you for being here. Thank you. Have a great day. Bye. And I welcome Dr. Ranganathan, who is on screen now, and I request Dr. Ananda Ganesh Raja to introduce our lead speaker, Dr. Ranganathan, who is alumnus of our faculty during his UG days and my own classmate. Please, Dr. Ananda Ganesh Raja, please introduce Dr. Ranganathan.
Yes. Ranganathan have been in the seed industry and in produce business for the last 17 years. Currently CEO of 7 Star Fruits Private Limited, part of Micro Group of companies, specialized in fruit exports to Europe and Russia, and involved in development of high quality fruit to type of disease-free planting material of horticultural crops like banana, apple, and cherry through micro propagations. We also are into fruit import and also have a direct to home delivery business. Fruits and vegetables were in farm fresh, hygienically sourced, and packed fruits and vegetables are sourced from the farm and delivered to the customer dining tables in major cities of Maharashtra like Mumbai and Aurangabad. Previously have held senior leadership and business management roles with seed industry giants like Myco and Nuziweed Seeds. Graduated in agricultural from Annamalai University in 1999 and did post-graduation in 2001 and doctorate from TNAU Coimbatore in 2004. Have also done 2 PG diploma in computer applications and bioinformatics from Bharatiyar University, Coimbatore in 2004. We welcome you, sir, for a lecture on residue management in table grapes. Thank you.
Okay, thanks a lot. I'll try to put up my presentation. Just give me a moment.
Dr. Ranga, whether you are sharing your slide, if it is any problem with your internet connectivity, we will be sharing.
Yeah, can you see my screen?
I think you are. I'm seeing your screen. Yes,
All right.
we are seeing.
Great.
Yeah. Thank you.
Okay. Yeah. So good morning all. So it's a great opportunity to kind of meet all of you, albeit virtually. So I don't do webinars. I send my R&D heads or agronomy head to do it. But this one particularly, I couldn't say no. Primarily for two reasons. One, talking to my own university where I learned all the trade, all the experiences. Am I coming through clearly? Are you able to hear me?
Yes, Ranga. You are
Okay.
audible.
Great. Thanks. So I think, so I think it's a great opportunity, as I said, particularly for two reasons. One, talking to my own university colleagues, and many of them being my friends and many of them being my own professors. So it's a thing that I didn't want to miss, number one. And number two, I think you are now the associate professor and my very close friend, Selva Muthukumaran. So I think, we call him very fondly Selva. So the only sincere person in our gang of friends, so I didn't want to miss Selva's request. So that is the reason here I am. And so I would have liked to come to the university. It's been 22 years since I left the university in 1999. So I would request Selva or maybe anyone in the team, the administration team, to just let me know in case I'm not coming through clearly, so that I know if there is a problem at my end in audio or anything. Please let me know that. So I think this, as far as I'm concerned, I think I manage. I'm going to talk about residue management. I'll keep my presentation short. When I was a student, also I didn't like lengthy lectures, so I will keep it very short. Maybe I'll take 20-25 minutes to complete my presentation, and then maybe I'll be very happy to sort of answer any questions that you might have. And I would like to allow the interaction with you more than my lecture as such. So I think Seven Star Fruits, the company that I head, is part of the Mahiko group of companies. You all might have heard a lot about Mahiko. It is one of the oldest in the private sector seed industry. So we started at the time of Green Revolution, in the 1960s. So I think now it's a very modern company. You might have heard about BT cotton, BT brinjal. The BT cotton transformed the Indian cotton industry as such, and BT cotton was brought to India by Mahiko. And now I think BT brinjal, which unfortunately as of now is not yet approved in India, but I think it's a very popular crop in Bangladesh, very close to our country. It's approved, and many more countries are very quickly going to approve BT brinjal. So, and we are hopeful that maybe in India soon we will get an approval for BT brinjal as well, and many more other crops are in the pipeline. So Seven Star is an horticultural arm of Mahiko. So all the agricultural crops we deal in Mahiko seeds, and all the horticultural crops we handle in Seven Star.
Keep with you. You make me charge and give you some printout of
Dr. B.R. Barwal, Selva, may I request that to be muted, please? I think probably at your end, there's a lot of noise.
No. Mobile, send my.
So Dr. B.R. Barwale, I think a lot of, he is a World Food Prize laureate. So he is the founder chairman of the company. So he was awarded the World Food Prize in '98, and are you able to hear me now?
Now we are hearing.
Yeah. Okay. So I think our founder chairman, Dr. B.R. Barwale, is the complete inspiration for the company. So I think he was a World Food Prize laureate in '98 and a Padma Bhushan in 2001. And all our programs, all our activities that we do, the basic mantra and the principles comes from this great man who actually passed away about 3-4 years back. But actually one is is inspiring us even to date, with this approach and the policy of farmer first. Our businesses, if you look at it in MyCode, I'll just give a brief snapshot of what we do. We are into inputs, which is seeds, seed treatment, chemicals, etc. We are also into engineering, in agricultural engineering. We have a company which focuses mainly on the silo manufacture and as well as storage solutions. And I run the fresh produce company, which handles primarily two things, the fruit exports and the fresh produce export and import, as well as we are into the planting material business. So I'll talk about it as we go along. So we have this three arm of businesses catering to the entire gamut of activities in this field. So some stats, I think Mahiko like to give a snapshot of what we do overall across the world. So we are an Indian multinational. We operate in across the world. We operate in Europe. We operate in East, other Asian countries like Vietnam. We have a significant presence in Africa. We also have our pack house and processing facilities in Zimbabwe, in Africa. So we sort of complete the circle as such in trying to provide the farmer what he needs. So there is mainly three arms of the business: the seed business, engineering, as well as the fresh fruit business in the company 7-Star Foods that I head. So basically, if you look at 7-Star as such, the focus crops for us in terms of export business is table grapes. We are one of the oldest exporters in India. We started way back in the 1990s. So it's one of the older companies in fruit exports primarily. But I think after I took over in the last 3 years back, we are more focused into the nursery side of the business. So I felt that there is it is like what cotton used to be, in agriculture. With very less interventions about, let us say, 20 years back. Horticulture has that kind of a scenario right now. So there is always a shortage of good quality, true to type and disease-free planting material in this segment. So that is where we are trying to cater now. So we are now into the micro propagation mainly. Micro propagation, all of you would understand, is you can make a true to type, disease-free, high-quality planting material and try the faster ramp up as possible in micro propagation, and that is where we are working on. We started with banana, and now we are working on apples and cherries, primarily meeting the micro propagation planting material requirement in the northern side of India, particularly in Jammu, Kashmir and Himachal. So we have many orchards there which are more than 100, even 120-year-old. So we are trying to replace those seminal orchards with good quality dwarfing rootstocks. So that is where our focus is right now, and all of our focus is on these two aspects of fruit exports as well as on good quality planting material. We have a lot of certifications to sell our material across the world, particularly if you cross India and beyond Asia, there is stringent requirements of quality. And there are various, the customers are very picky because they have high purchasing power, but at the same time, they are very picky when they select their material. One is, of course, the residue that I'm going to talk about, but other than the residue, even the overall, the quality of the product, the way they like their fruit to be when they pick up from their retail shelf, they're very picky. So we need to make sure that we pass the stringent quality requirements of various certifications that are required to sell our fruits across the world. So one of them being the Global Gap to sell in Europe, various other Rainforest Alliance again to sell in Netherlands and so on. So we have all sort of gamut of certifications, which I think our pack house, our cold chain, as well as our business processes are inclined and have cleared all these quality requirements to sell our material across the world. Some of the brief of the product that I'm going to talk about today, I've selected table grapes primarily because I think this is one of our bread and butter in terms of the fruit that we handle, but it is also one of the fruits that is actually the #1 in India in terms of exportable commodity, if you look at horticulture as such. So I think there's the total production is around 3,000 tons in 30 lakh tons in India. So I think in terms of, if you look at, we have various varieties. The most prominent one, which is in terms of exportable material from India, is Thompson Seedless, which is the most popular variety that goes. You have the temperature, these requirements that you are seeing on the right side of the slide basically are our exporting requirements. The fruits undergo a process called pre-cooling, which I think is a part of the cold chain infrastructure wherein you need to cool the material, cool the fruits Okay, I'm seeing some hand raised. Would you mind just note down your questions and maybe we can take it up at the last, if it's okay with you. We can take it up all. As I said, I'll complete it in 20 minutes and maybe I can answer the rest of the points at the last. So Maharashtra being the hotspot in terms of fruit exports in India, I think particularly for grapes. In Maharashtra, there is a district called Nasik, which is the primary sort of a hub wherein the exportable grapes are handled. You have table grapes also in Karnataka and even in Tamil Nadu as well. But the bulk of the exportable activity happens in Maharashtra and particularly in the district of Nasik. Like I said, Maharashtra being the number one, it handles about 63% overall. And this is the biggest chunk of exportable activity that happens, and that happens from one state and one district. These are some of the varieties that are popular in India, just to give a brief idea of how the variety-wise breakup happens. So Thompson Seedless, as I mentioned, is the king in terms of the export. In terms of the sweetness, it is slightly less sweeter compared to, let us say, a Sharat or a Sonaka Seedless. But in terms of how the European taste and the Russian taste is attuned to it, they like this sort of a grapes, unlike the Indian palate. So Thompson Seedless is the major chunk. Almost 90% of the exports that happens from India is Thompson Seedless. One of the other aspects in horticulture, which is very important, is the rootstock. Because the rootstock determines the height of the tree. Rootstock gives the disease and the pest resistance to the tree. So primarily, the selection of the rootstock is very, very important when a vineyard is designed. So in India, the most popular one is the salinity tolerant Dogridge rootstock, which is the most popular one, but it's again an older one. You have many new rootstocks coming up, which are occupying bigger spaces, particularly in the vineyards of California and South Africa. But in India, Dogridge takes the lion's share of the rootstock, and it is a very popular one. Now, coming to if you look at the export-import basket of grapes as such, as I mentioned earlier, grapes is the largest in terms of the exportable fruit that happens from India. So 2.4 lakh tons is a huge number of grapes get exported. But again, if you look at the overall scheme of things in terms of the overall production, again, it is less than 10%. So, and import, there is a new trend in India now, particularly in major cities like Mumbai, wherein the purchasing power of people going up, people particularly, and the health conscience, particularly with the advent of COVID, you would see a lot of focus on being healthy and eating a lot of fruits and vegetables, particularly this craze on improving our immunity levels. So you would there is a lot of interest in people picking and choosing the fruit varieties which are not normally available in India. And now with the advent of new retail shelves, there is a high level of premium retail outlets, mushrooming in various cities, particularly cities like Mumbai and Bangalore. You will have new outlets, and these are focusing on high value fruits. And you would have hear a lot of name like let's say an avocado. This is one of our focus crops, what we are working on right now. And even in grapes, people want new varieties to taste. So you would see a lot of Californian varieties being imported. So this is a trend, even though we have so much of export availability and the foods being in spite of there is so much consumption happening domestically and still a huge volume of 2.5 lakh tons being exported. You still have, you are still seeing over the last 2-3 years, there is a significant increase in imports of particularly grapes from the United States and from California, primarily of new varieties, which are more sweeter, which give a more tardy taste to the customers, different sense of taste that they're able to experience. So this again gives a snapshot of what I just mentioned in terms of the export, even though it is the highest volume. So it is again, if you look at the overall scheme of things, this is less than 10%. Now, just to give a brief snapshot of the season. So I think, the peak season normally is February, March, wherein the Thompson Seedless comes to harvest, and bulk of the fruit exports happen during this season. Now, if you look at the European markets, one of the main advantages that India has is our prices, particularly in terms of manpower. I'll talk about it even further in the presentation, is comparatively low to other, let us say, the European countries or even the American ones. So that is one of the significant advantages which India has, which I think India has been focusing on for the last almost 15-20 years. Now, Netherlands, UK, and Germany form the bulk of the exports, bulk of the imports. They are the major customers of the Indian grapes. And in Asian countries, you have other Middle Eastern countries as well as Bangladesh being the customers. On an average, you will have seasonal fluctuations happening, but on an average, about 10,000 containers. A container is a 40-feet container which can house about 20 tons. So now on an average, about 10,000 containers gets exported to Europe. If you look at the in terms of Indian rupees, is about a 2,000 crore kind of a business. And particularly Netherlands and Germany have fruit requirements or fruit in demand in their retail shelves throughout the year. Customers are, like I said, the purchasing power is high. There is, they want the high quality and premium grapes to be supplied. Price is not the issue as far as the European customers go. So if you look at the ports as such, wherein I think the entire, exports are handled, Rotterdam in Netherlands, there is a port in Netherlands called Rotterdam, which is sort of called the gateway to Europe, because that's the first entry of all the exports that happens. And as far as India is concerned, the most busiest port in terms of the export handling is the Nhava Sheva port in Mumbai, which handles the major exports that happens from Mumbai, from entire India. So Europe, UK, and Canada, and again, UK customers pay the highest for Indian grapes. So UK and the Canadian customers pay the highest for Indian grapes. The most premium prices that the exporters are able to get primarily are from UK and Canada. But again, the flip side to it is they require the most premium ones in terms of the quality as well. So let us say in a total overall area that we have, even less than 1% get shipped to these countries, because the most premium ones, the A+ grade as we call, goes to UK and Canada. But again, they fetch a very high price. So this gives a snapshot of what I just talked about in terms of the quantity, Netherlands being the major one in terms of the inflow. But again, the port of Netherlands, the material that enters Netherlands also goes into various other Schengen countries in Europe. Now, some of the idea for the quality standards, to talk about the business side of the exports, you want loose bunches of 400 to 600 grams. You want damage-free, scar-free material. The berry sizes have to be 16 to 18 meters. The soluble solids, or the TSS, or the bricks as we call it, which is the sugar, the sweetness, the minimum has to be 17 to 18 bricks in terms of the sweetness for the material to get exported. So coming to the pest side, you have many pests in grapes, right? But as far as the exports are concerned, which I think as an exporter, which worries me the most, if you look at it in terms of the pests, or the mealy bugs and the thrips, as well as the stem borers. Now, I have not listed, the anthracnose in terms of the disease here, along with the downy mildews and the powdery mildews. To just give a snapshot of these are the 4 or 5 that I think both pest and diseases that we are concerned with. You have a lot more that I think will be more of an academic interest, but I have listed only those which I think. Just give me a moment. Which I think I am concerned as an exporter to give a snapshot of 4 or 5, which I have highlighted here are the major concerns. Now, what we normally do is, in terms of some key practices that the growers adopt. Now, I'll talk about the pest management as I go along, but one of the key things is very important in grapes is the pruning. Pruning is removing the leaves and allowing the lesser competition and for the berry size to improve. So the pruning is a very common practice in all the horticultural crops and most of the trees, right from apple to grapes. This is a very common one. In grapes, there are 2 types of pruning that happens. One we call as the foundation pruning, or otherwise it's called summer pruning. In the rural language, they call it as summer pruning, which is done in April. Wherein I think after the harvest, after one month after the harvest, they retain one basal node and then remove everything. And then the other one happens, which is called as a quality improvement pruning, or the winter pruning, as the farmers call it. This happens to activate the dormant auxiliary buds that are dormant during the winter, to activate them. This is mainly done to get loose bunches, which are very much essential for exports. And one of the key things in pest management, like I said, residues being a major concern whenever exportable grapes are handled. So one of the key things that the farmers are always advocated in exportable grapes, and these guys are very much experienced. So you will see the grape growers have been doing this for the last 15, 20 years. Like I mentioned, grapes is one of the first fruits to start getting exported from India. So these farmers are almost experts. So we have a process, and every exporter has its own team of people, agronomists as well as the entomologists, to visit the farmers and guide them on the practices that they need to adopt and when to stop spraying, to ensure that the residues are not left out before harvest. There is a very minimal amount of spraying is advocated, and only up to the 50% blooming stage. Most of the exportable farmers follow the integrated IPM methods and also the cultural management methods, right from for mealybugs or for that matter use of biocontrol. There is a very minimal spraying that happens, and even the spraying that happens only up to the 50% blooming stage. Beyond that, there is very minimal activity that happens as far as the spraying is concerned, particularly because, as I said, the residues, if it is left at the time of harvest, it decides there is a big difference in terms of the prices that a farmer gets. Let us say the difference is chalk and cheese. It's a huge difference in terms of the material being sold for domestic consumption or the material that is getting exported. So, and the farmers are always advocated to avoid fungicides like mancozeb or thiram near to the harvest, particularly because I think these leave the residues which are highly carcinogenic. Now, some of the molecules of concern are, these have listed out the molecules which are very common, in India. If you look at, material like carbendazim or chlorpyrifos, these are very common chemicals that are used. But these are all molecules of concern, which I think have been highlighted by the European supermarkets, which I think the farmers are very aware of. So knowingly, unknowingly, domestically, when we consume grapes, there is a high level of chemical load on those materials. But when the material goes for export, you will see that there is very minimal load, particularly because the European markets are very, very strict on the residues. Every country in Europe, has a different residue level. I think these residue levels before the exporting season are published by that particular country's phytosanitary authorities. And these are very, we as an exporter, we need to make sure that, we follow each and every country's residue level requirements. And only when we clear it, the material gets exported. And these are, even if the material gets exported without following that, once the material reaches the customs of those countries, the testing is done. And if it is found out that I think, the material has residues, a very strict penalty is implemented on the exporter. And there are chances of the exporters getting banned as well for a few years. So all of the exporters make sure that the banned chemicals and the molecules of concern in terms of the residue are very strictly adhered to. Now, I'll just give you a brief snapshot of for any person who doesn't understand how the grape exports work. Every grape grower who wants to send his material for export has to register his material, register his plot with APIDA. APIDA is the regulator for the exports that happens from India. So, and there is a standard guideline that you can also go into the APIDA website and see. There is a standard spraying schedule that every farmer has to adhere to. And he has to keep a complete record of all his spraying schedules. So all the exporters, so what is our role in this? So we the exporter, once the farmer is registered with APIDA and his plot is registered, we register the growers, our own growers who are wanting to supply material to us and guide them as in the farm management. As I mentioned, throughout the season, our team visits the field and guides the farmers as to what practice they should follow and what they should not follow. I think this is a complete, right from the planting till the material is harvested, our team is constantly on the ground. We are also involved in various farmer welfare measures, like, providing some stationery to their children and, doing some activities for the women in the family and all that. I think that is a continuous engagement that an exporter has to do with the farmer. So either the exporter does contract farming or it's an open farming wherein the farm is picked before the season. The grower registration is a mandatory activity that we do. And once the material is ready to be harvested, during the harvest, the samples are taken by our quality team. And then what we call as the MRL testing or the minimum residue level testing. You have APIDA has certified certain labs in India who are qualified by APIDA to issue the MRL certificates. We call as residue certificates in our export term. So these residue certificates, only when issued by certain APIDA certified labs, is valid for exports. So we exporters send our samples that are drawn by our quality team to these APIDA certified labs. And based on the residue certificate and the residue available in that particular plot, then export destination is decided. For example, if it qualifies for the European residue limits, the material is shipped to Europe. If it is not, then there is some lenient, some countries that are very lenient, like Russia, for example, in residues. So then we decide that that material has to go to Russia. And if it doesn't meet any requirements, that the particular plot is rejected. So this is the process that happens. And once the material is identified, the material is harvested, packed at our, we have a pack houses. Every exporter has there is a standard infrastructure that I think is required for packing the material. And I think we need to make sure that I think, the cold chain as well as the pre-cooling requirements are fulfilled once the material is packed. So, and once the material is ready to ship, we need to get a phytosanitary certificate, which is issued by the state government authorities. The phytosanitary inspectors of the government visit the pack house, see the material, check the residues, and then issue what we call as a phytosanitary certificate. Once this certificate is issued, the material is ready to be shipped to the port and from there onto the sea, to the destination. So normally a transit time for Europe is about 40 days. The material is in the sea, and it takes about 40 days to reach Rotterdam. And in case of certain Russian ports, it is a shorter transit time. For example, in Russia, you have two ports. One is called Novorossiysk, the other one St. Petersburg. So these are the two prominent Russian ports. These involve a lesser, shorter transit time compared to what we have in Europe. Once the material reaches the customs there, and the importer, you have the importers who import the material from the exporter, and these importers run their own residue level tests to meet the European guidelines. Only when the material is cleared of that, the material is then moved to their pack houses and then eventually to the retailers. You have huge retail chains in Europe, like they are called, one is called Aldi, which is like our Big Bazaar and Reliance. You have huge retail chains in Europe. The most prominent ones are Aldi and Rewe. These are huge retail chains which you will see across the European countries, which they are very high premium chains and very, very stringent on the quality. Once the material, they run their own tests of quality and residue levels before the material is given to their customers. And only when the residue limit is as per the prescription, the material is then sold to the customer. The retailers put the material, the fruits, in their retail shelves. So one of the issues with this entire business, what I just explained, the process, the farmers are paid in advance when the material is lifted. But I think the entire risk of the customer and the importer paying us, or the retailers, the retail chains in Europe paying us, happens only after the material is sold. So as an exporter, you take a huge investment risk until the material gets sold. And again, what we call as the entire business is on a consignment basis. That is, you send the material on your own risk, the material reaches. We need to make sure that the material is safe in the sea till 40 days. The material safely reaches there, convinces the importer, and also convinces the retail chains. Only after the customer is happy, they pay us. So every exporter runs crores of rupees of investment risk while the material is being shipped, but that is the nature of the business. So I think as a grape exporter, you always take that risk. And some of the issues of, what I think India has faced in the past in terms of various residue issues in 2019 primarily, and even in 2015, many huge number of containers from India were stopped midway as they were in the transit to Europe, particularly that there was a concern of a chemical called chloromethylene chloride, which was a banned chemical at that time in Europe. So which is a very common one here. And similarly, in 2019, the popular retail chain called Rewe in Netherlands, they had, by the time they had detected that there was a residue issue of a particular chemical, some of the fruits were sold to the customers. And in Europe, the biosafety is a very, very serious issue, as well as the food safety particularly is a very serious issue. And any customer can, sue the retail chains and make millions of euros of damages. It is very common in Europe. So I think Rewe made a public announcement wherein there was a concern, and they made an announcement saying that, the customers can get their refunds, and they publicly apologized as well. That particular, fruits shipment was from one of the exporter in India, and the exporter was banned for 3 years. Similarly, Greenyard, a very popular importer in Netherlands, which is a supplier to Rewe, had to fight a lot of food safety concerns, particularly due to the Indian grade. So there is a general perception in Europe that the Indian farmers sometimes compromise the quality or the residue levels to get more yields. So I think there is an always on pressure on the Indian exporter to make sure that the fruit that he provides, that he supplies to his customers in Europe, are residue free. And I'll just one more slide I'll talk about before I close. So just to give a brief snapshot of how grape exports in India and why I think we have certain strengths compared to other Asian countries. One of the things that India is blessed with has, it's a very agroclimatic diversity that this country has. You are having an exportable, you can grow exportable grapes in Maharashtra, you can grow it in south of the country in Tamil Nadu as well. So this is one of the strength, and agroclimatic diversity within the state is blessed, which I think very few European countries are blessed with. Second, we now have more and more, high-end technologies available, that can be used to get higher yields. I think you will constantly hear NRC, the National Research Center of Grapes, that is there in Pune, come up with new advent of technologies which can be used by the farmers to increase productivity. And one of the things that other point that India is blessed with, the time of harvest of what we can get in India, like in the January, February, March, no other European country or South American country has that particular time of harvest. So that is one of the reasons why India is able to ship this kind of a quantity to Europe. And again, the most important thing which reduces the cost of cultivation in India for the farmers for grapes is the low cost availability of manpower. Grapes again is a very labor intensive crop. So, and one of the reasons why, Europe is not able to match, or for that matter, even the United States or other Californian growers are not able to compete is this. The manpower cost that is there in India is low. This is one of the significant advantages that India has as far as exports are concerned. One, if you look at the opportunities as such, Europe has been the primary area for India, and it has been there for the last 1.5 decades. The challenge that comes is Europe is reaching a peak in terms of the requirements from India. And every year there is so much of competition, particularly in terms of Indian grapes, that every Indian exporter is competing with each other to sort of pull. There is what we call as the competition to the bottom, and everyone is competing with each other to reduce the prices and the quality. So the overall quality of Indian exports is going down. So the need for the Indian government as well as APEDA to look at new export horizons. And one of the countries that has very recently allowed Indian grape exports is Australia. But again, there is a huge distance, between India and down under to Australia. And, particularly countries like United States don't allow Indian grapes to protect their own growers of California. So I think this and even the other American countries in the American continent like Peru. So these countries compared to the Californian growers, Indian growers cannot compete in those countries. And there is a very stringent quality standards in certain countries like Canada, wherein most of the Indian growers are not able to compete or not able to meet sort of the Canadian requirements and are not able to supply our fruits there. In terms of, in terms of the opportunities, in terms of the weakness and the threats, if you look at it, one of the things that are happening primarily is, the climate changes that are sort of you are hearing every day, and you are reading every day about the climate changes that are causing, huge, the wildfires in California, right up to, the heavy rains in Nasik in Maharashtra. Every season, the last 2-3 years, there have been very high rainfall, particularly just before harvest, due to which the quality of the harvest has gone down. So this is one of the major concerns that I think, even though it is not in the control of either we as a businessman or the farmers, this is one of the key changes that are impacting the competitiveness of the Indian farmer, and which the policymakers have to take into cognizance. And the other one being the political angle to it. See, there is a high level of subsidy that is given to the exporter as well as to the grape farmer to involve in grape exports. And this is very politically sensitive topic as well. But one of the prime concerns, what happens is, if any person who is, let us say, into real estate or into other fields, right, only during the export season comes into the grape exports just to get that subsidy that is being given. So what happens is for organized players, people who follow their quality and people who make sure that the quality fruit goes, are competing with these players who have very low overheads. Now, for example, for a company like us, we have a huge team and huge infrastructure and huge overheads that we incur. But you have a person who can source any other pack house available on take on lease and then export. But once the quality is not meeting the standards, the overall quality of the Indian grapes gets a bad name. So these are some of the areas that I think now the Indian government has identified that particular issue and are now trying to reduce the subsidy to make sure that I think, the fly-by-night operators don't come and ruin the business and ruin the name of the country as such. So these are some of the pictures of the activity that happens during a pack house during the grape export season. You will see a huge number of people there. This is actually a photograph taken pre-COVID. So I think during COVID, I think we are allowed to run the facility with only 50% of the staff. And in Maharashtra, the kind of impact that COVID caused last year, right during the export season. And for the last 2 years, the lockdown was announced in the last year in March, when there was a peak export season happening. And then again, this year, during February, March, I think the second wave came in and Maharashtra went for a complete lockdown. So I think this is one of the areas, one of the constant worries that we have. And in my own team, there were a lot of issues with COVID, and we had to tread very, very cautiously and reduce the team strength to make sure that I think the facility itself runs and the export happens. So these are some of the challenges that we have, which I think the whole country and in fact the whole world face, not only us. So this is some of the challenge that you need to meet. And again, COVID also caused most of the Europe lockdowns. So much of our customers in Europe and also the retail chains were closed. And right from you ship the material and the material is already in sea, and suddenly Europe goes into a lockdown. And you have invested at least 30-40 crores in that, and then you get the money only when the sale happens. So many of our exporters lost our sleep for a few months during the COVID lockdown and as well as during the second wave. So some of the nature of the business, I guess. Thanks a lot, and I'll be happy to take any questions that you might have. Thank you.
Thank you, Dr. Ranganathan, and I request the participants to pose their questions through chat box.
Okay, I'll try and answer some of the questions that I have got. One, I think Arul Pattaswamy's question. He asks, what are the problems in exporting agri commodities to other countries apart from insecticide residues? Well, I talked about some of the concerns, Arul, in my presentation. And one of the things is the stringent quality standards. Quality standards doesn't necessarily mean the insecticide residues. If you take a plot, let us say on average grape plot, you get a 20-ton yield per acre. Out of that, only about 4 or 5 tons will be good enough to export to Europe. So for an average farmer, you cannot, the rest of it goes into the domestic consumption. So, so what we eat as day in, day out as Indians, what we eat grapes, or let us say are the rejected ones that are rejected for export. That is what we eat every day. So, I would urge all of you to, before you give the grapes to your kids, make sure that they are washed 3 or 4 times. So that is how it is. It's very, it's a chemical intensive crop. And, but at the same time, once the farmer gives it for export, like I talked about the challenges, and then again, there are very few countries who allow Indian grapes. Like I mentioned, the Europe countries does, but again, the American ones, like I mentioned, the qualities are very, very stringent. Even the UK standards are very, very stringent. So the quality standards in terms of the size of the fruit, as well as, the overall appeal, the bricks are very different to each country. And that is the major challenge for the farmer too. sort of follow and practice. And the other question that I think he asks is, I need clear idea about the subsidy. Well, subsidy actually, you will hear a lot of grape exporters say that, they want subsidy. Now, as an exporter, as an organized exporter, I say that I don't like subsidy. Simply because I think, subsidy allows every Tom, Dick and Harry to comes into this business, right? The thing is, you need to be serious about grape export or fruit export for that matter. But I think subsidy, you just shouldn't do the business just for subsidy. There is to just give a snapshot of what was subsidy. The earlier subsidy was 5%. That is, whatever you realize from your export proceeds, you get 5% of that as subsidy. This is this subsidy. So once you get the realization from your importers, 5% of that you can claim from the government as subsidy. Now that subsidy has been reduced to 3%. While I think my other exporters are very angry about it, I'm actually happy about it because, even though my inflow in subsidy will go down, this will make sure that, the only organized people are doing this business, which will overall increase the quality of Indian exports to Europe, and the bad name that has been created that Indian exports are mediocre, that image will go. So that is more important from a longer term perspective. Okay, I think there are a lot of career questions, which I think I wouldn't like to answer on a call. But I think, what we can do, Selva, is maybe I think, what kind of an opportunities that we have, etc. I think, we can take that up maybe separately. But I think one thing that I'll tell you is, if you are good in what you are doing, if you have what we look for as an industry is practical knowledge, right? So I think, the syllabus alone will not teach you. What you need to look at is more of the, take a keen interest in what you are doing, right? Rather than only, the marks that you score or what we call as the OGPA competition, right? I think you need to have that mindset to come out of that OGPA mindset, because that OGPA that you get is not what the industry is looking for. Because at least the interviews that I do, I never look for how much OGPA a person has scored. I look for what sort of a practical knowledge that the person has, more than what OGPA he has. So that is what is important. So whatever you do, just look for, develop a deep interest to it and do a deep dive instead of rote learning. I think that will be my only suggestion as a friend and as an advice as a probable senior.
So, dear Dr. Ranganathan, I take this opportunity to share your success mantra, a little of it, so that our students get motivated. Although it is out of the context as of today, but if you still you share your mantra, success mantra, that will be a motivation to these youngsters, because you also from this very campus. So we did our undergraduation together, but still the long travel that you took and you reached almost a pinnacle of being a CEO of a company, right? This seems to be a motivational dream story, right? So for them, if you just share a few thoughts, although it is out of today's context, I accept your views, but still, if you just talk a few minutes about that, that would be nice, because your extempore talk will always motivate our students. Right? As to, I do remember your college days and your presentation in the classroom for LAC culture. I still get that into my memory always. So if you just share your insights on that, it will be nice. Sorry for deviating, right?
No. Anything for you, Selva. You asked me to sing, I will do that right here. He's a very close friend of mine. So I think anything for him. But look, I think, see, like I said, one of the things that we need to, I always tell, any junior that I encounter or my own kids, the things that I tell is, whatever you do, right? It's the most innocuous thing to say, but it has a lot of meaning, right? Whatever you do, you try to do a deep dive into it. Right. One of the things that we always compete when you are in college or when you are doing an undergrad, or for that matter, when I did my PhD, we are into what we call as a bio bubble. You would hear constantly people calling COVID bio bubble. You would see the Indian cricket team in a bio bubble. Similar to that, as students, you are in a bio bubble, right? The thing that it is like a pupa talking to an entomologist, right? It's like, you are there, you are sort of in a cocoon, right? Very, very, in a comfort zone. And the only thing that you need to know now is how much you're going to score and, how much OGPA I'm going to get. See, the world beyond is much more than the OGPA. Right. So what I will encourage you to think, encourage you, all the students, you might be in graduation, you might be in post-graduation. One of the things that you need to look at is, what can I explore deep down into the subject that I'm looking at? Or what is the implication of this, what I am learning in an industry, right? If you look at it, there is no need for you to ask, what is my scope or where will I fit in? Right? There are many people waiting to hire you, right? So that is one of the things as an industry or any company for that matter, that as a value addition you can provide. Because that question you will get asked in whatever interviews that are going to do simultaneously. The first question that I ask whenever I hire is, what sort of value you will provide me? If you tell me, look, I have scored 95% or 9.5 OGPA, I don't care about it, right? I'm going to say, what value you are going to provide to my company, or you are going to add to the company. So any subject, it might be entomology, it might be pathology, it might be extension, any subject you do, what is important is, more than the theory, come out of this bio bubble that we are in as students, right? And try to make sure that whatever you are doing today or what you learned on that particular day, what is the implication or the practical application of it in the industry? If you learn that, as long as you are going to be in academia, that is fine. Right, that I think you need to choose that, right? Now Selva decided to be in academia, and he's a great academician. I respect him for that. Now any person, now I decided I'm not a good researcher, even though I did my PhD. I always felt, I think that is not my calling. I need to go out, right? And I felt that industry is my calling. Now many people still ask me, then if industry was your calling, why didn't you do PhD? Why did you do your PhD? Selva would know the answer. I wanted to be a doctor, right? And I missed my marks by 0.5%. And then I had to come to agriculture. Agriculture was never my choice. It happened by chance. And just to get that doctor before my name, I did my PhD. Nothing beyond that. So I never wanted to be a researcher. So, so that is one, you need to decide what you want to do. And, you decide and you go for that. Whatever you do, if you do 100% or give 200%, like I give my example to my kids about Selva being, many of the things that we learned from him and whatever little I know about entomology is from him. And, so this is one of the things that, whatever you do, give your 200%. I'm sure this is my success mantra, right? It is not, I'm not going to, this is not some kind of a Steve Jobs talk that you're going to put it in a book, right? It's a very simple mantra that I can tell you. Whatever you do, give your 200%. I'm sure you will be successful.
Thank you, Ranga. And there is one interesting question about the residues while they will end up in wine making. What is the possibility of the residues in grapes ending up in wine?
Okay, no, I think, see, these are completely different segments. The varieties, the way a vineyard is designed. India is, in terms of winemaking, there is a very, very small opportunity in India in terms of winemaking as such, and the kind of varieties that we have and the kind of climate we have. You have very small, and you would have heard about a popular winemaker called Sula, S-U-L-A, which is very popular wine that again comes from Nasik primarily, which is a very most popular Indian wine. But again, the winemaking as such in India, if you, and any wine drinker, wouldn't prefer Indian wines because you have much better, French wines available. If you are really a wine aficionado, or you like, wines, I think I would recommend, even though it might be wrong to say or unpatriotic to say, India is not a great winemaker. And because of the climate that we have, and because of the varieties that we have, so it's a completely different segment altogether. And these two have to be looked at it completely differently. And, the way it is winemaking is managed, you have very, that is a different subject altogether. And maybe I will talk about winemaking and some other presentation. But this, table grapes and wine grapes shouldn't be confused. These are completely different segments or completely different market opportunity.
And another question, handled pesticide residue meter. Is there any like that? I hope it is not so.
No, it is not. The pesticide residues are not measured by hand. Your bricks can be measured by hand. If you have seen the gallery of photographs that I showed, you would have seen a bricks meter there. So sugar level can be checked by hand. But I think now I think we are working on using drones to do a lot of these other quality parameters check. You would have seen drone being very commonly used. Now I think even drones are now being used for pesticide sprays to see the the size of and attributes of the fruits, the berry size and all that. But I think even the drones cannot test you the residue levels. The residue levels have to be properly checked in the lab, in a lab condition, take samples, and there are very clear guidelines. For APIDA has clearly listed out the guidelines for taking an MRL sample. How many samples have to be taken at what level of the field? To give us a simple layman understanding, 4 samples at different parts of the field have to be taken, and then so that you do a proper testing in the lab, wherein because by a handheld device there is a lot of, even if I develop one tomorrow, the chance of error, human error in terms of the machine error, even the configuration error is very high. And the residue levels in many European countries are lowered to the level of 0.01 mg per kilo. So even if you make a small mistake, the entire shipment will get rejected and you will lose crores of rupees. So I don't think anyone will take that kind of a risk in this.
Thank you, Ranga. And actually, from my side, the impact of residue in your export, whether you treat residue as a most important problem in export or anything other than that.
See, to me, I think the biggest challenge there's a concern for all of us is the MRL levels or the residue levels, and as well as the physical damage that the fruit encounters. See, the transit time, like I mentioned, is 40 days in the sea, and what we call as there is a temperature recorder that is put in each consignment, which is a standard operating procedure. We call as temperature recorders. There are 2 temperature recorders are put in every container while the material is packed. But what happens is when the material is in transit, the control of the material that we have as an exporter is seized the moment it leaves your pack house. Beyond that, the material is in control of some marine line. So we all make sure that we go for the best marine lines available. Like you would have heard about the name called Maersk, M-A-E-R-S-K, is one of the very popular shipping lines and a very premium one. We make sure that we go for the best shipping line available. But then again, it is up to them to manage the container till it is delivered to our customer. If the temperature of the customer is not maintained during the transit, it's being a highly perishable item. The fruits being a highly perishable item immediately gets spoiled. And by the time the fruit reaches, there is a high level of rejections that happen in the fruit. And any shipping line, is only giving you insurance. You can take marine insurance. The marine insurance will only allow you a return or a compensation if the material, if the sea, if the ship sinks in the sea, or if some cyclone happens, right? Then you get what we call as the acts of God. If such things happens, you get a marine insurance. But if the fruits get spoiled, you don't get a insurance for that. So this is one of the prime concerns that we have, how the containers are being managed, while the material is shipped. And one other issue that I want you guys to understand is the marine, the transport costs involved in this business are huge. And, particularly the last 2 years when the vaccines due to COVID were getting, shipped. And, when the Chinese containers were banned in many of the countries, and, most of these containers, the big traffic happens from China. And when the Chinese containers were banned and, due to COVID, and many of the ports in China were stopped, and as well as the vaccine manufacturers were wanted to, and every country gave them emergency authorization. So all these containers were locked for, vaccine, because there was a national emergency. So, and, the fruits and other things can wait. The people were dying. So the importance was the vaccines to come. So the containers either were not available or the prices of these containers skyrocketed. So for every exporter, the margin that we earn in this business were almost next to zero. So these are one of the challenges that, the business happens, particularly when your customer is sitting thousands of miles away from you.
Thank you, Dr. Ranganathan. And this, if there is no more queries being raised up, I think we have answered all, right? Okay. So thanks for accepting my invitation, because we know how busy you are. Being a CEO of a company, it is not easy to accommodate yourself for an hour, right? And that too under this pandemic, after the lockdown, everything is opening up, and you will be hectic looking after the business, and you will be taking care of, and you will be in charge of everything, right? Being a chief executive officer, it's not an easy job to do, right? I thank immensely for accepting our invitation. And before being proposed, before the formal vote of thanks being proposed, I put forth another request with due, right? All the things that come up my mind, and I'm putting you a request again, right? If there is, please look into the possibility of arranging for some campus interview for the graduates who have been coming out here, and probably we will be looking into the modalities. Probably I will be in touch with you, so that if there is a requirement, I will be in touch with you. I hope there are some requirements. You will acknowledge that, I think, with the same gesture that you shown towards us, right? I hope we will also be in a position to invite you for an offline event soon after the COVID situation eases out, right? It will be a nice meeting again, right? I hope
I'll be happy to. I'll be happy to do that, Silva. Like I mentioned, I think it's been 22 years since I stepped to Chidambaram as well as to the university, and I keep telling my kids stories about the Kurunji hostel and the Malligai hostel and all that. So I wanted to show them around the facility, and it's been a long time. And I think my wife also keeps on telling me that I think she has to go to Chidambaram to the temple and all that. So maybe sometime I'll definitely bring them along, and it will be happy. I wanted to come to the university as well and meet all of you people. So many of my very good friends are there, and many of the professors that whom I respect. I don't know whether people like Dr. Selvanarayan and Dr. Arivudanambi are still around. I mean, convey my regards to them. So I think there's a lot of professors that I've learned a lot of my trade from. And I think I still remember that LAC seminar, and Mr. Selvanarayan used to be my hero those days. So, and I hope he is doing well. So I think, all of these memories are still there with me, and I'll be very happy to visit the university whenever given an opportunity.
Thank you, Ranga, and we are most happy to host you again, right? And I request Dr. Ananda Ganesaraja to propose formal vote of thanks to Dr. Ranganathan, right?
On behalf of Department of Entomology, I thank Dr. S. Ranganathan in his lecture on residue management in grapes. In his lecture, he narrated on challenges in grapes marketing and insecticide residues issues. Further, he pointed that various insecticides used on grapes and their residue level. So really, it was an excellent lecture which motivated our students. Thank you, Doctor.
Yeah, my pleasure to the entire team. Take care, stay healthy, all of you. Thank you. Thank you, Silva.
Thank you, Dr. Ranga, and see you. Bye. Thank you. So we are coming to a conclusion of the technical session, first technical session, and we will have our second technical session scheduled by 2:30 in the afternoon. So we have two speakers for the session 2. One from both of them are from the UPL, and first one is Dr. Luigi, who is a global technical development manager. He is from Italy. And another one is again our own alumnus, who is a national lead, technical lead insecticides. Right? So he is housed in Coimbatore. Dr. A. Prabhu is my senior and is classmate of our Dr. R. Kanagarajan. Right? He is kind enough to accept our invitation. All these invitation has been shared only just a few days before, and they readily acknowledged and they accepted and to be here. So we will be winding up the session 1 now, and we will be taking up the second session 2 by 2:30. All of you can join in the same link for all the technical sessions in both the days. The link is same. You can join using the same link, and today afternoon you are most welcome by 2:30. Right, your participation is most solicited, because without you the conferences or the workshop is nothing. Right, only with your interaction it gains more importance. Thank you for your patient participation now in the technical session 1. I expect such continued cooperation in other remaining 3 sessions. Thank you.